I started my career actually at CFRI, more or less. I remember it's over 30 years now that I gave a presentation here when I was a postdoc. So let me just guide you through all the good times that I had as a scientist in the cystic fibrosis field. So basically, let me just introduce you quickly to the history of cystic fibrosis and where I came in. So in the 1946, it was discovered that cystic fibrosis is inherited. And then a couple of years later, then Hans Ussing developed a technique called the Ussing assay that's still used today. And that's a fantastic discovery that Hans Ussing did. And he worked on the frog skin, and he developed this Ussing model where he described the mechanism for active sodium absorption in that time. So mainly the sodium was absorbed and excreted at the basolateral membrane. So that mechanism, that basic ion transport mechanism that we still think of today for sodium absorption, was developed in the frog skin. So then 30 years later, Paul Quinton discovered that cystic fibrosis is an underlying chloride ion channel defect. And shortly after that, the CFTR was cloned and discovered on chromosome 7. So then in the beginning of the 1990s, I came in, I had a postdoctoral fellowship to study in the US. And back then I studied actually also on the frog, and I studied frog lung. So what I did as a graduate student, I was studying potassium channels in the basolateral membrane of an epithelium, and I studied this in the frog lung, which is really an interesting model because you can just cut it open and fold it and put it in an Ussing chamber. So then I was a postdoc in Terry Mackin's lab at UC Berkeley for a long time. And in that lab, we discovered that CFTR is also conductive for chloride and bicarbonate. And one of the major discoveries we also made that we could stimulate CFTR by cyclic AMP independent mechanism. And that was the first discovery that brought us to this molecule genistein that suggested that there are mechanisms for small molecules to be activators of the CFTR ion channel. And so back then we discovered together that with Jonathan Widdicombe at the Children's Hospital in Oakland that we could stimulate CFTR by the small molecule genistein. And this also worked in patients. And the importance was that it worked in these patients with the G551D mutation that had some residual CFTR function in the membrane. And that basically provided the underlying rationale for CFTR drug discovery later on. Then I had the pleasure of working with the late Dieter Grunert, and who developed, also together with Jonathan Widdicombe, airway epithelial cell lines. And with Dieter, I was able to establish the CFBE41- cell line and the 16HB cell lines as a robust model to study ion transport for cystic fibrosis. And so later on 2012, we had this first CFTR modulator drug approved. And after that, I had the pleasure of working with Dennis Nielsen and Megan McGarry on serotyping individual cells from cystic fibrosis patients. And now more recently, I started to get into lipid nanoparticles and the delivery of messenger RNA. So today's talk is about the preclinical assessment of CFTR function in children with rare CF genotypes. And I'm giving you examples of 5 patients that we examined in terms of their chloride ion transport function. So today we know that there are approximately 105,000 people from 94 countries who have cystic fibrosis. So that number increased considerably over the past years. And then we have an additional number of undiagnosed cases, so that we have approximately 53,000 people in the whole world who basically are not diagnosed yet. And so this population is here in Argentina, in India, in some parts of Africa, and Egypt, and Turkey. So I think there is still a lot to do in terms of identifying all these people. So let me just quickly introduce the cystic fibrosis gene. So we all know cystic fibrosis is a monogenetic disease. It affects one gene. However, there are by now more than 2,000 mutations that are found in the gene. So let me just walk you through how the gene looks like. So it's a huge gene. So it has 189 kilobases, and then it has 27 exons, and these intronic regions are these lines in between. So now there are then 2,000 different mutations somewhat distributed in the gene. So we have this homozygous F508del mutation, that's the most common mutation. And then we have other ones like stop mutations, missense mutations that span the entire region here of the protein. So what's important now to understand when we come and go about nucleotide-based approaches to correct CFTR, we should be aware that we have 4,440 nucleotides that we have to somehow package into our delivery system in order to deliver the CFTR mRNA. And so later on, then when the CFTR mRNA is translated, it ends up in a protein that has 480 amino acids. So the using assay that's been developed and heavily used in my lab is the gold standard for the preclinical assessment of CFTR function. And so here, that's an example how this chamber looks like. So it's a plexiglass chamber, has a little slider in here, has 4 electrodes. That's usually right is my apical side, and the other one is the blood side, the serosal side. And so we can put in tissues, for example, and I think that's going to be interesting because we talked about sinus tissue this morning from Matt Forius. So we had sinus tissue here from Peter Wang's lab from Stanford that we could actually measure. And we're measuring ion transport in these tissues. And then here is, for example, a cell culture from a cystic fibrosis patient. So these are cells that we are growing, basically a patient in a dish model. So when we do an ex vivo analysis of CFTR function in people with CFTR mutations that are less common, we first isolate the cells by nasal brushing, and we then have to expand these cells that we brush. And there's now significant advancement in the expansion protocols. We have then to culture them, put them in the using assay, and then we do our protocol for the assessment of CFTR function. And so let me just explain something just fundamental. So here's the short circuit current, the chloride ion current plotted over time. And so now for the efficacy of CFTR modulator drugs, there is actually an important convention. So we basically inhibit the sodium current, then it goes down, then we stimulate with forskolin, and then with the CFTR modulator, in this case a VX770, and then we block with the inhibitor. So what we use then as a normal function is basically the forskolin stimulated portion. And that's usually set at 100%. So what you find here is basically a description of the 5 patients that we examined in terms of CFTR function. And all these patients, and I'm not sure if and the last patient was a 4-year-old with a very ultra rare mutation. It's a CFTR deletion mutation. And I'm going to explain that one in detail. So you can see now when we talk about different mutations, it really can get confusing. And so we have 2,500 mutations, and I was just lucky to have a sample here from different rare mutations. But at this time, we really didn't know, do these mutations or do these patients respond to the modulators or don't they do it? So, and I think during that time, it was kind of important to identify which patients would be responsive. So now in order to do this, we were rushing cells and we put them in a medium called EpiX medium. And that was actually a very important advancement because the EpiX medium allowed us to grow a lot of cells with very simple methods. So it was not radiation involved or anything. So we can see here that it took about, the cells were just growing much at higher quantities compared to the conventional medium. And so this propagation medium that we used was then able to generate cultures that we could use in using assay. So I don't want to go too much into all these descriptions, but I think what is important for maybe a parent or a CF patient is really to understand your mutation. And I think it's getting so complex with all the different mutations. And just sit down. I always talk to my students, where is the mutation? Just go into the nucleotide sequences and look where it is. And then it just provides so much more information and sense, and you get a feel where you are in the CFTR molecule. So we can do that now for each mutation. And I think what is also then, in addition, very confusing is we have the intronic sections, and these intronic sections also give us some trouble sometimes. So we have, for example, this common R117H mutation that resides in exon 4. And then we have these little 7T nucleotides that can vary. And it's then a polymorphism that basically determines if you have a severe manifestation of CF or a less severe, or the 90 does not cause any symptoms. So we also have then to look at these intronic regions and understand what does actually 7T mean. So now with this particular CFTR genotype, we had a F508del in combination with the C850-dub-A. And so that's also a mutation that creates a stop codon in exon 7. And it's basically then shutting off the channel, and it will not be functional. So in this particular patient, we had this R334W mutation that's located in exon 8. And it has been reported in a few patients, and it has a higher prevalence in Portuguese and Brazilian populations. And it is also thought to be somehow associated with some bicarbonate activity that's higher in these mutations. And so this one was paired then with another splice mutations, and it is particularly a mutation that is found in the Hispanic CF population. And so in this particular genotype, we didn't really know how this genotype would respond. And so let's come to the last one, to the large deletion mutation. And I think what is really amazing about this mutation is that it basically knocks out the entire CFTR gene, because it is deleted at this between exon 2 and 3, and then it basically creates a premature stop codon, and the CFTR is basically not translated anymore. And this is a mutation that's particularly prevalent in the Eastern Europe population. It's also now found in Algeria, Egypt, Cyprus. So it is a very common mutation that's found in Eastern Europe. So and here is how we do the measurements in the lab. So first we determine the quality of the cultures by doing a transepithelial resistance measurements that gives us a value for the fitness of the epithelial cells. And so all of them kind of are lying in this upper area between 600 and 1,000 ohm times centimeter square. And there are some in this deletion mutation that resulted in a less quality culture, so we were not able to use some of the inserts. But what I plotted here is what we're basically then measuring is the chloride channel activity, the ENaC activity, and the calcium activated activity. And so this is then just demonstrating that there are certain levels of CFTR function that we can measure. We can attribute a certain amiloride sensitive activity, and then also see if there's a functional calcium activated channel. And in particular, with this one CF patient, we did not induce a calcium activated chloride channel on activity, and it was rather going into the opposite direction, which could be that in this particular patient, there is something going on with his calcium activated chloride conductance as well, or this could mean that there is just a maybe potassium secretion. That's another factor that could contribute to it. However, I think it gives us a good quality of a sense of the quality of the cultures that we're generating. So what is also interesting is that we plot here the chloride current versus the sweat chloride concentration. So it nicely groups in these cultures. So we have a CF positive patients with a sweat chloride that's larger than 60 millimoles. And these are all these mutations that fall into this group. And then as expected, we have the R117H7T mutation in combination with delta F is an intermediate sweat chloride concentration between 30 and 59 millimolar. And then here our negative control, our non-CF subject is CF negative when the sweat chloride drops below 2029. So in here, I wanted to show you in particular the results we obtained for the deletion mutation because it was very unexpected. So here's just an overview about the experiment. So we add amiloride, we block the sodium conductance, and then we add forskolin, VX770, and then surprisingly we do see some small but detectable CFTR inhibitor effects, and those are blown up here a little bit. So we treated these tissues or these cultures. Either they were untreated, they were treated with VX809 overnight or with VX661 overnight. And although these cultures are, or these currents are so small, we were able to detect an inhibitory effect of the inhibitor. And interestingly, these cultures responded better to VX661 compared to the VX809. So this gave us some thought to think because we didn't expect this result, and we checked the literature a little bit and found that this particular mutation was found to express some truncated messenger RNA, and that paper by Dirk was published in 2000 and never followed up in terms of function. So I think that we had now some evidence to suggest that this particular mutation, when it is even in a homozygous patient, can perhaps be stimulated with the modulators. And there is also some confusion in the literature. And I wanted to point that out because in this particular classification scheme, what you see here, so that's the deletion 2,3, the 21 kilobases that are missing, was actually found to be classified as a class 7 mutation, and then it was reclassified again because of the messenger RNA. So although it says here unrescuable, I think there is still some portion that may be available to rescue. So and here's a nice, another summary how we plot these patient in a dish data. So we can think about each individual now with a certain genotype. And I think it makes sense to just sort them here for the, here we have a lot of currents here in the R117H heterozygous patient. And then we can basically always identify using VX770 responsiveness, I would call it. The responsiveness to VX770 clearly shows then inhibitor responses to CFTR in inhibitor 172. So in all these 5 patients, we can detect some degree of stimulation by the modulator, by the VX770. And there are also some genotypes, and I wanted to point those out. These are some stop mutations and splice mutations that really do not respond to the VX770. And so this really tells us that the Using assay is really a valid assay to discriminate between responsiveness and how we will benefit or not from modulator treatment. So then our next focus is, so when we have these particular genotypes and patients associated with it, so they cannot be, or they're not treatable with CFTR modulators. And I think that is the whole point where the gene therapy comes in. So in these patients, or at least in these patient-in-a-dish models, I would say when we see some responsiveness to VX770, they are likely to respond in vivo to some degree. And again, what is important then so how do we make a cutoff? And currently the cutoff to qualify for CFTR modulator treatment is that in these assays, we need to have at least 10% normal CFTR function. So if you go through this line here, we can basically determine which genotype or which patient behind that genotype would actually respond. And then we have these cases where it's borderline. And so that's where we actually do not know, should they get the therapy or actually not. And with this particular F508del patient, it was interesting because I thought, this response is not really robust for this F508del patient. And interestingly, so when I talked to the clinicians, they were telling me, yeah, this particular patient also does not respond well to Orkambi therapy. So these data would suggest that there is some other mutation maybe, or we don't know yet why this FDEL homozygous patient is resistant. So now let me quickly touch base with the genetic medicine. And I want to get really, I know that's the end of the talk, get to the end of the talk. So let me tell you one thing. So there have been clinical trials that delivered messenger RNA. And the first clinical trial was started around 2017 by Translate Bio. So they were actually applying their messenger RNA packaged in a lipid nanoparticle to CF patients. The trial was called Restore CF. And then it was ended, I think, sometimes after 2020, because it didn't show efficacy. And also because patients developed some side effects in response to the therapy, like fibro responses, fever, and cough. And basically, it was not well tolerated. But I think that was still a trial that was very informative. And now there are 3 more trials that started to enter the clinical phases here in a phase 1 or 1-2 phase for safety and efficacy and tolerability. And these new trials are all done in healthy subjects. So there is one that's done by Moderna and Vertex, one that's done by Arcturus, and the recent one is done by Recode. And I expect that we see the results soon, but these are, as I said before, healthy controls, healthy subjects. So now let me just quickly, we still have some next generation scientists in the audience. So for them, this is going to be a piece of cake. So it's basically the concept of delivering mRNA is, this time the mRNA doesn't come from the nucleus, okay? So it comes from the outside, and we are packaging the mRNA into these lipid nanoparticles, and they have to enter the cell by endocytosis, and then finally the mRNA needs to be released from the endosome and then translate into a peptide or into a protein, basically. So it's here at this large unit of the ribosome where the messenger RNA gets translated into the amino acids. And so then we know here's a table which amino acid is encoded by which nucleotide or by which nucleotide triplet. So you may notice that there are some amino acids, they can be encoded by 4 different triplets. I mean, that's all high school 101, I guess. But I think we really use this information now in order to optimize, for example, the nucleotide sequence of the CFTR in terms of stability of the nucleotide. So now here comes my question for the next generation. So what is the first amino acid of the CFTR protein? Does anyone know? Jeff? Methionine, yeah, exactly. So to be honest with you, so I had to ask also ChatGPT. So the first amino acid of the CFTR protein is methionine, which is encoded by the start codon AUG. Okay, take notes. In the gene sequence, methionine serves as the initial amino acid in the synthesis of most eukaryotic proteins. And here's the structure of it. So now, I think sometimes we don't trust ChatGPT, or do we? Okay, so I double checked. And so I went here into the sequence from the NIH, and so, yeah, it is M. Okay. It's M, it's methionine. Okay, so one thing you remember from my talk, okay, it's just M, methionine, first amino acid. Okay, so now let's get back to the clinical trials and how we deal about getting messenger RNA across the membrane and basically being translated into a protein. So I think we heard that before we saw that slide. So the LNP formulations that are currently used for making LNPs, and I recently learned that from my collaborator at UC Berkeley, Neil Murthy. He basically invited me to his lab, and I could really create these lipid nanoparticles. So there's a test lipid, and that's either an ionizable lipid or cationic lipid. It enhances the endosomal escape, so to release the messenger RNA from the LNP. Then there's a lipid called DOPI. It's a helper lipid. It's supposed to encapsulate the cargo. Then these LNPs have cholesterol to promote stability. And then they have this DMP-PEG lipid that anchors the lipid in the membrane, and it improves the biodistribution. And now recently we have a 5th component, and these are then the short LNPs that have certain signals attached that bind to certain proteins. And so we generate these lipids, these LNPs, and then we add the messenger RNA to these lipids, and we can do that actually just by hand pipetting. It works. And then we create this LNP and mix it then to the cell cultures. And so my idea was that, you know, we generate the lipid, the LNPs, like in the clinical trials, and we're going to mix them to the cell culture medium and see what's happening. So the ultimate goal then was to identify under which conditions do we see actually expression of a protein when we add it to epithelial or to bronchial epithelial cells. So we first used the enhanced GFP for expression, and that's a very small protein. So it's only 239 amino acids versus CFTR is much, much larger. And so we used the expression of this gene and pipetted the LNPs either to 96 well plates or to a transwell plate. So in the importance of this assay that I wanted to develop was we can basically add the LNP formulation to subconfluent cells, or what's happening when we have a more in vivo situations. And that's basically mimicked by the transwell, where we have cells grown as cell monolayers. So the cells are forming a tight epithelial layer. And here are the data. So what we generally, or what saw was when we added the LNPs to cells that were grown in a 96-well plate, they were grown on plastic, they were just not totally confluent. Our lipids or our LNPs, and that's the GFP expression that resulted from it, was pretty good in these cells when they are in these subconfluent cells. And as soon as we added the LNPs, the same LNP to a tight epithelial monolayer, we don't see the expression to occur so robust anymore. So here we have like 70 to 80%, and in the transval plate where the cells are tight and electrically tight, we only see maybe 5 to 10%. And so we tested a whole series of lipids, and to just show you a little bit the difference again, that's subconfluent cells, and they really express high, so after 48 hours. And here's the transval experiment where we have one cell here, and we have about 20 cells lighting up after 48 hours. So it is more difficult than I thought. And then it's also something to be careful when reading the literature. I really want to know the detail. How were the cells actually treated? So were they really adding the LNPs to a confluent monolayer, or is in the literature more or less you use like this optimization by just transfecting cells that are not non-confluent? And so I think the delivery is basically the issue at the moment. If we want to do or develop protocols, how do we screen our LNPs for expression of genes? And that needs improvement. So even though we sometimes can have a low expression or a small number of cells who do express the CFTR gene, for example, I'm coming back to some of the work I did with Peter Gruner, and we were actually looking at different populations of cells. We were mixing cells. So we are mixing the 16-HBE cells, the CFTR expressing cells that normally express CFTR. So that's our normal control. And then the CFPE41 minus cells. And these are the CF cells that are homozygous for the F508 del. And interestingly, so here is basically the cells that are lighting up are the cells that are expressing these are the normal cells. And so with the increasing number of normal cells, we get more and more CFTR current that we can measure. And interestingly, so we saw that even at if we have 1% of normal cells in our cultures, we still see significant increases in the current, which means that even a small number of cells can express the CFTR in like a normal CFTR expressing cells, and still will be detected by the Osing assay. So 1% wild type cells generated about 5 to 10% of the normal CFTR activity, which is quite promising. And it is promising because we know that 50% of normal CFTR gene expression is required to maintain a normal phenotype. That's what we know from the heterozygous patients. But then there is in this literature the other dogma that we only require probably less than 8% based on these individuals who had an in-frame deletion exon 10, that we would need 8% or less of bronchial CFTR transcript in order to be able to have a normal phenotype in the lung. So and that's actually also where the 10% cutoff comes from. So now in summary, I showed you that the EpiX technology is a viable method for cell culture expansion for CFTR functional assays. So we had a remarkable reduction in the starting material as low as 20,000 cells. And that's something that we can achieve in a really in a simple nasal swab on children without anesthesia. So it's like almost you're doing a COVID test. And therefore we could do this as this nasal brushing technology now in children as young as 3 years old without topical anesthesia. And then we found out that these particular genotypes, the F508del and the C850AA, as well as the splice mutation and the homozygous CFTR deletion mutations, were responsive to CFTR modulators with a response that was larger than 10%. And the conclusion is that the CFTR function assay allows for phenotypical classification of rare CF genotypes in children. So what is the outlook of these studies? It would assist additional people with CF whose variants are so far not included on the FDA drug label to petition for an off-label trial period of the CFTR modulator therapy, and we encourage steps for official FDA label inclusions. So where variants are found in people of color and other minoritized populations, extending the drug eligibility to additional CFTR variants will be a critical step in addressing the racial and ethnic inequities in CF care and outcomes. And the preclinical studies for the CFTR mRNA delivery to CF bronchial epithelial cells are expected to perform better in non-confluent cells and suggest further validation in confluent cell monolayers. So I'd like to acknowledge all my colleagues here at the UCSF Benioff Children's Hospital. I had the pleasure of working with Jonathan Middicombe, Horst Fischer, and these were students, Francis Kyles in my lab. And then UCSF, we had Walter Finkbeiner providing us the cultures, Dieter Grunert also providing us with CFBE cells. And yeah, I just wanted to now thank Paul Quinton and for being in the Department of Pediatrics now at UC San Diego, and my new collaborators, Niren Murthy at the Innovative Genomics Institute and at the Stanford Consortium. I'm expanding now with CF research with Mike Wilkinson. And last but not least, I want to thank all the students in my lab, and I particularly want to thank the Elizabeth Nash Foundation for their generous support to support my lab with the summer student internship program. And these were students back in Oakland that I worked during the summer. And some of them had cystic fibrosis, some had a family member with cystic fibrosis, and they brought really cystic fibrosis so much closer. When you are a bench scientist, all of a sudden I understood what it means to have cystic fibrosis, and it really changed my career and how I think about it. And so this is now my new setup in San Diego. So I already managed to get my chambers up and running there. And that is only possible with CFRI support. And I'm so grateful for the support. And here are two new students from UCLA that I had the pleasure to work with and train and educate the next generation of graduate students and undergraduate students. So it was, yeah, a fantastic year. And I really want to thank you for bearing with me. It's late right now. And thanks for your attention. And thank you, CFRI.